Hello everyone, how are you all? Welcome to One Page Biology. In today's video, we are going to discuss another very important and beautiful topic. The name of the topic is population genetics. Now this topic is very important because every year from this particular topic there are questions asked in CSIR NET Life Science examination as well as in different PhD level entrance exams like GATE, TIFR, even in the SET examination. So let us understand this concept of population genetics. Now, first of all, what do we understand by the word population? So, in simple words, a population is large group of individuals. Now, whenever we talk about individuals, we can consider here any particular type of organism. It can be a group of bacteria, it can be a group of birds, it can be a group of humans. So, precisely whenever we talk about population genetics, here we are considering a group of individuals. And in the group of individuals, we study certain characteristics of that particular individuals. Now, in genetics, you already know that the information of characters is present inside the genes. In simple words, the identical copies of genes are called as alleles. So, what are alleles? Alleles are basically the identical copies of genes which are present on the homologous chromosomes. So, by understanding the characteristics of population, we can study its evolution. So, like for example, let's say there is one population A and there is one population B. Population A has got dark red color eyes and population B has got dark brown color eyes. So, we can study that over a period of time what is the change in the characteristic of population A with respect to their eye color. We can also study the same in case of population B. So ultimately, whenever we study about the changes in the characteristics which are ultimately coded by their respective alleles, we can study the evolution of a particular population. And that is the major aim of studying population genetics. So it is very important whenever we study population genetics, we have to understand the allelic frequency of a particular character. So in this particular video, I am going to teach you how to calculate allelic frequency of a particular population. If you know how to calculate allelic frequency of a population, it will be easier for you to understand and study the nature of that population, the characteristics of that population. So as a genetist, it will be easier for you to study the evolution of that population. So let us understand how to calculate the allelic frequency. So let me give you an example of a particular tiger moth which is specifically called as a scarlet tiger moth. Now this particular moth has got spots on the surface of its body. So this particular moth has got different types of genotypes. Let's say one particular type of genotype is capital A, capital A. The another type of genotype is capital A, small a, and one more type of genotype is small a, small a. That means capital A are nothing but the homozygous individuals of this particular moth. Capital A, small a are heterozygous individuals of this particular moth, and small a are homozygous recessive individuals of this particular moth. Now, I'm not telling you the phenotypes. Let's just assume that capital A is majorly coding for a particular phenotype number 1. Capital A small a is coding for another phenotype, and likewise even small a, that is homozygous recessive individuals, are coding for a particular phenotype. Now let's say a total number of homozygous individuals of this particular scarlet tiger moth are nothing but around 452. Then the heterozygous individuals of this scarlet tiger moth are around 43, and the homozygous recessive individuals of this particular tiger moth are 2 respectively. Now we have to understand that in this particular population of scarlet tiger moth, there are around 452 individuals who have the genotype of capital A. Then there are around 43 individuals which have the genotype of capital A small a, and there are 2 individuals which have the genotype of small a. So if we have to first of all find out the frequency of individuals which have capital A as their genotype, how we will find? So first we have to know what is the total number of individuals of the scarlet tiger moth. So let's calculate the total number of individuals. So if we calculate the total number of individuals, it is nothing but 452 plus 43 plus 2. So this will roughly come around 497. So now if we have to calculate the frequency of capital A, that is, now we are calculating the frequency of homozygous individuals, that is precisely homozygous dominant individuals. So what will be the frequency of homozygous dominant individuals? So it will be the total number of individuals which are having capital A as their alleles divided by the total number of individuals. So here the individuals having capital A as their genotype are nothing but 452, and the total number of individuals are 497. So if you calculate roughly, this is going to be around 0.909. Same way, if we have to calculate the frequency of small a, that is the frequency of homozygous recessive individuals, how we will calculate it? It will be the total number of individuals having small a genotype divided by the total number of individuals, that is 2 divided by 497, and that is going to be equal to 0.004. So now it is clear that the frequency of homozygous dominant individuals is 0.909 and the frequency of homozygous recessive individuals is 0.004. We can also calculate the frequency of heterozygous individuals. So the frequency of heterozygous individuals is going to be, again, if you see the heterozygous individuals were 43 in number, so it will be 43 divided by 497. It will be coming around 0.087. So that means in this group of scarlet tiger moth individuals, the frequency of homozygous dominant individuals is 0.909, the frequency of homozygous recessive individuals is 0.004, and the frequency of heterozygous individuals is 0.087. Now everyone, you have to know that this is the frequency of individuals. This is not the frequency of alleles. So we are yet to find out the allelic frequency. So let us calculate the allelic frequency. So now when I have to find out the frequency of, let's say, allele capital A, because capital A is the dominant allele. So in individual there is going to be either two capital A alleles, that means that individual is going to be homozygous dominant, or the individual will have one capital A and one small a, that means the individual is heterozygous dominant, or the individual is going to be small a, that means that particular individual doesn't have any of the dominant A alleles. So if we have to calculate the frequency of capital A in the population, so how can we calculate it? So it is going to be, frequency of capital A will be frequency of total number of homozygous dominant alleles plus half the frequency of heterozygous individuals. So now the frequency of homozygous dominant individuals was around, if we see, it was 0.909. So the frequency of homozygous dominant individuals was 0.909, and the frequency of heterozygous individuals is nothing but 0.087. So this will be divided by 2. So this will be around 0.909 plus 0.087 divided by 2 will be roughly around 0.043. I'm just taking a rough calculation. So if you calculate this, so this will be approximately around 0.952. That means the frequency of allele capital A is nothing but 0.952. Here you have to understand, why did we take the total frequency of homozygous dominant individuals and half the frequency of heterozygous dominant individuals? The answer is, the homozygous dominant individuals are having both the copies of capital A and capital A. So that was around 0.909. So it was the total frequency of individuals having capital A as their alleles. At the same time, the heterozygous individuals have only single capital A in their genotype. So we have to divide it by half, because the total frequency of heterozygous individuals was around 0.087. So we cannot directly take 0.087, because 0.087 is the total frequency of one dominant allele capital A and one recessive allele small a. So it is a total frequency of heterozygous individuals. So hence we have to take the half frequency of heterozygous individuals, because here we are only calculating the allelic frequency of capital A, which is the dominant allele. So that's why the formula is total frequency of homozygous dominant alleles plus half the frequency of heterozygous alleles. So now this has come to around 0.952. Similarly, if you calculate the frequency of a recessive allele, that is frequency of small a, it will be the same formula, but only that in this case it will be frequency of homozygous recessive alleles plus half of frequency of heterozygous alleles. Because again here in this case, the heterozygous individuals have only a single copy of a recessive allele. So this will come to around, if we calculate, what was the total frequency of homozygous recessive allele? It was around 0.004. So it will be 0.004 plus the half the frequency of heterozygous individuals. So we just calculated it, it was around half the frequency was 0.043. So plus 0.043. So this will be around, so this if we calculate, it will be around 0.047. So now we know that the frequency of capital A is 0.952 and the frequency of small a is 0.047. So the frequency of capital A here we can indicate it as P, and the frequency of small a we can indicate as Q, right? So if we calculate the total frequency of allele, that is the total frequency will be P plus Q, that is going to be 0.92 plus 0.047. So it will be approximately 0.967. So roughly it is going to be around 1. That means the total frequency of alleles in a population is always equal to 1. Just now we saw that total frequency, that is P plus Q, was approximately around 0.96. So that means it is approximately around 1. So in this particular way, we have calculated the allelic frequency. There is one more method by which we can also calculate the allelic frequency. Suppose if you have to directly find out the frequency of capital A, there is a formula for that also. How can we directly find out the frequency of capital A? The formula for that will be 2 multiplied by the total number of homozygous dominant individuals, that is capital A plus the number of heterozygous individuals divided by 2 into total number of individuals. Let's calculate that. So 2 into the total number of dominant individuals were, if you recollect, it was around 452. So 452 plus the total number of heterozygous individuals were 43 upon 2 into total number of individuals were 497. Now remember that why are we taking here 2 into total number of individuals? Because since we are calculating the frequency of dominant allele A, it is nothing but 2 into number of homozygous individuals. Why 2? Because here we are considering the number of individuals. So obviously the homozygous individuals are having twice the number of dominant alleles, whereas a heterozygous individual is having only a single copy of a dominant allele. So that's why we are multiplying a homozygous dominant individual by 2. At the same time, in case of the denominator, you can see total number of individuals multiplied by 2, because in the denominator we are taking the total number of alleles. So we know that every individual has got 2 copies of alleles, so it will be 2 into total number of individuals. So if you calculate roughly, this will be around 0.952. So same way, so if you just compare, in the previous case also we got the frequency of capital A as 0.9, and in this case again we are getting the frequency of capital A as 0.9. Same way we can also find out the frequency of small a, and that will be 2 into the total number of homozygous recessive individuals plus heterozygous individual upon 2 into total number of individuals. So this will be around 2 into 2 plus 43 upon again 2 multiplied by 497. So this will come to around 0.047. So you can just compare it with the previous frequency which we had calculated. Again, here also we got the frequency of small a as 0.047. In this case also we are getting the frequency of 0.047. So either ways you can calculate the frequency of the allele. One method is by taking their frequencies, that is finding out the frequency of homozygous individuals, heterozygous individuals, and then just by adding the frequency of homozygous individuals plus half the frequency of heterozygous individuals, you can either find out the frequency of allele, or the other formula directly, as we can see here, that is by finding out the number of individuals having that particular respective allele. That is, in case of, if we have to find out the frequency of a dominant allele, then we can find out by the formula 2 into the number of homozygous dominant individuals plus the heterozygous individuals divided by 2 into the total number of individuals. So always remember that the frequency of any of the allele, whether it is a dominant allele or a recessive allele, it is going to be less than 1, because obviously not all the individuals are going to have the dominant allele, neither the individuals are going to have a recessive allele. So always the frequency of a particular allele is going to be less than 1. But if you calculate the total frequency of alleles, that is the frequency of capital A plus the frequency of small a, it is going to be around approximately 1. So this is very important as a genetist to find out the allelic frequency, because after finding the allelic frequency of a particular character, we can study the evolution of that character in a population. So in this video, I hope you have understood how to calculate the allelic frequency. So I hope you have understood this particular concept of the population genetics. If you have understood this particular concept, please like the video and share it with your friends. If you want me to make a video on any other particular topic related to biology, please let me know in the comment section. See you in the next video with some another biology lecture. Till that time, take care of yourself. Thank you so much. Bye bye.